to pick one of these?
Yeah, could we fly past those, please?
That's great, thank you.
Do you think that these are carnivorous sponges again?
Maybe. If we take a look at the stalks, we can really tell.
Do you want to zoom in on this?
On the spicules, yeah.
I think
If not, it's all right.
It's okay.
I'm a little close to that. I think
No
I'm
worries, no
let's
worries.
keep going.
past it with the light.
Let's go.
What do we look for if we're trying to determine whether it's a carnivorous sponge or not?
The carnivorous sponges are distinct usually by the tops of the creature, and so they can have like parasol shapes, or they can be kind of like spiky. They can have lots of spicules lined on the sides. They really try to maximize the surface area. A lot of the other sponges, like these glass sponges, kind of look smoother. They're made of glass, so you can kind of see the
that way.
way that their spicules are formed. Just looks a little bit different texture on it. Wider osculums on the glass sponges because they are passive filter feeders, so they still need to draw the water through, whereas the carnivorous sponges don't necessarily.
Okay, go for zoom.
It looks great, thank you very much.
Have you gotten the chance to see any of the carnivorous
We can
sponges?
do a flyby that one, unless you're being pulled to this side, in which case don't worry. I think it's another crinoid anyway. Let's zoom in on that.
On the one in the center screen, or where?
There it is.
Over there. Go for zoom.
There you go, okay.
Would you look at that?
Good.
Holothurian.
Thank you, that's unusual. I don't know how to make the highlights clip, that one.
Yeah, that's definitely going to be a 5. Okay, I think I'm going to head out soon, so I have some science questions I've been saving up.
Oh, yay.
So John, you were talking earlier a little bit about why the nodules might be kind of on top instead of mixed in. I don't know if you've said this a million times, but how do the nodules get formed? Like, I understand when there's a crust, you know, it's like the metals are precipitating out of the water and falling down.
Yep, no, that's a good question. So you're familiar with the carbon cycle, right? So there's, because of course that's integral to Earth's climate, and so there's carbon in the air and carbon in the water and carbon in rocks and in biological material. And so pretty much every element in the periodic table has a kind of cycle.
Can you zoom on that?
And when it comes to the ocean, the actual seawater, that's great, thank you, we'll keep going. It can be turned into a concept of residence time. So some elements are happy to stay in seawater, and the best example of that, of course, are the elements of sodium and chlorine, right? So sodium chloride forms salt, and it'll stay in the sea forever, pretty much. And other elements like to drop out straight away, or fairly quickly. Two of the elements that like to drop out are iron and manganese, and their residence time is, it's still not instantaneous, but it's thousands of years kind of thing before they get an excuse to drop out for some physiochemical reason. Yeah, maybe we'll just have a quick check on that on the way past. So now you've got this iron and manganese that's being put into the oceans, either from rivers coming off land or from hydrothermal vents deep in the earth, on the seafloor, and it drifts around in the sea. And after a certain period of time, it'll get the opportunity to drop out, and it can form any kind of crust or any kind of mat, whatever. It's all fine. If it is being dropped out into a place like this, which is perhaps the most common form in the whole world, it'll start to grow concentric balls. So it'll find a tooth or a piece of mud or even just an old piece of ferromanganese, and it'll coat it. And it'll slowly coat it, and then every now and then that gets turned over or upset, and so it'll grow around it like that. And if it's not being moved, or if it's quiet enough, it'll form a crust. And we found a few crusts even on this traverse. So there are places where you can see it's not being turned enough, but there's some kind of mechanism turning them. And we know that from dating of them, and we know that from the textures that we see in different parts of the ocean. That's got to be the case. What exactly turns them? Well, I might have mentioned before that there's all these different processes at different speeds, and nodules are the slowest. Just go look at that. Slowest growing of all of them by orders of magnitude. So like nodules grow 1,000 times slower than sediment accumulates on the seabed, for example. So if you kind of made a movie at which you could just see the nodules growing slowly, it'd be like a blizzard of sediment landing on them at the same time. Yeah, maybe we'll pick one of these. Which one of these three do you like?
Let's go to
Sandy?
the two on the right, please.
These two? Right.
Okay.
So, yeah. And what you can do is, if you cut a nodule in half, you can see these growth rings, and then you can try and date them, and you can even look at the chemistry of the rings. And so the leading hypothesis based on a combination of dating and the physical chemical conditions of the growth was put out by some folks at the BGR in Germany.
Zoom there, please.
And what their conclusion is that each ring is an interglacial epoch, basically. So it's 50 to 60,000 years apart. Yeah, there's two exactly the same thing. And the Earth's entire oceans have changed slightly in chemistry, going from fully oxic to suboxic conditions. And that means you get a different iron manganese mineral forming. There you go. That was a bit of a long answer, but does that help?
Yeah, that's cool. Thank you. So I guess here are the currents stronger than they might be in some places that just have crust? Or is it still, they're not really sure why nodules formed here versus crusts in other places?
Yeah.
Like what's moving them around more?
What you can do is you can list all the processes at once. And there are places where we know currents influence the growth of nodules because we find nodules grown in certain ways. It has to be either microcurrents or sometimes currents that go along contours of hills. But I think it's probably the amount of biological activity. So this is extremely low biomass, even though the biodiversity is high. And I think there's just some places that just don't get turned often enough. And maybe once a crust starts, it won't get turned anymore. It's just too big or something like that, in which case it'll keep growing. Yeah. The crusts can go on rocks, they can grow on sediment, they can grow on top, they can grow inside the sediment. Every combination has been found.
And I guess the sediment that gets trapped inside there, is there still like live bacteria or does it kind of fossilize, you know?
They are highly porous, these ferromanganese minerals, and people have been trying to decide if they're chemically grown or biochemically grown. And as far as I know, the science is still not totally resolved. So people find textures in there, and some people say that's a bacterial mat texture, and other people say, no, that's just the way the chemistry would grow anyway. From the reading I did a few years ago, I haven't seen anything recently. One thing I do recall reading is that if you take a nodule and you do some eDNA on it, you'll sequence a series of bacteria that live in the nodule. And then if you take a sediment from right next door, you'll get a different, there's some overlap I presume, but you'll get differences in species. So there are definitely nodule endemic bacteria as well. So but there must be certain types of bacteria that particularly like the chemistry or the conditions in nodules.
Cool.
I can't recall where the paper was, but I remember reading something about that. And that's such a simple test. You know, you'd give that a lot of credibility.
Do you guys have an estimate of like how many millions of years it took to form these nodules?
So we don't yet have enough, let's go look at that, enough dating in the Cooks. But based on nodules done in other parts of the world, it's usually of the order of tens of millimeters per million years, so maybe 20 or 30 or 40 down to below 10. So the kind that are below 10, so 4, 5, 6 millimeters per million years, seem to be similar to these. So what we call hydrogenetic nodules. So we've assumed it's the same, but we've not actually collected our own dates.
Thank you.
Yeah, and if there's anyone in the chat who wants to date a nodule for us, we can certainly supply some samples. And we'd be happy to work with someone who's got the time and the equipment to do that. Yeah, we'll zoom in on this one, please.
Okay, zoom.
Yeah, great.
And go wide, please.
So what's our depth? Oh,
51.95. We're still not
no.
quite there.
Looking at the bathymetry map, this is where we had our best chance to get to 5,200.
Right. It's just teasing us like 2 meters away.
Yeah. Well, we are going to go over a very gentle ridge, and then
Okay.
I think we might have a chance on the other side of that.
Maybe that'll be it.
If we don't get to the next half hour or so, I wouldn't be optimistic.
Lindsay, did you really just come in here to be present for that moment?
I actually did not know that this was maybe going to happen during this watch, so
Oh.
I'm excited. Will that be like the deepest that the Nautilus has ever had an ROV go to?
Little Hercules, yeah.
Wow. Cool.
Atalanta has gone a little bit deeper. Battle of Midway Atoll in 2023, but not with Little Hercules.
Cool.
Yeah. Well, hopefully cool. I mean, yeah, we might get there.
Well, I think we hit the record anyway. It just wasn't 5,200 meters.
Yeah, I know. We want that like round number.
Thanks, Tim.
Should have ample opportunity to smash past 5,200 at the next dive site, so
Oh.
don't sweat it too much.
Do you have some intel that we don't?
Yeah, tell me so. I didn't know that. Oh.
Okay, I'm going to do another question. This one's maybe more for Matt, but John was saying earlier that like sonar technology isn't quite there yet to like be able to tell if there's nodules underneath us or whatever. I was wondering if the multi-beam or any of the sonar on the Nautilus is good enough to tell if we're like there's a sandy bottom or like a hard bottom or anything like that.
I can definitely do that.
Yeah.
Yeah.
Yeah, sure. So we're talking there about backscatter or echo intensity, basically. So you get a couple of products back when you put a pressure wave out into the water, an acoustic pulse, and it comes back. You get the two-way travel time, which is what we're looking for to give us our depth. And then you're going to get the intensity of the echo, which is basically power out, and then you subtract power back. That is what you've got, and you can account for various forms of losses while that energy is propagating through the water column and back up to you. So there's quite a bit of, I'll say, I think even people who do this for a living would admit that the math can be a little fuzzy sometimes, just because there is quite a lot to account for. But we can get a very reliable relative measure of the intensity of the echo coming back from the seafloor. And a good rule of thumb is that the more energy you get back, the harder the bottom is, the less energy you get back, the softer the bottom is. And when you pair that with, you know, the gold standard is kind of an ROV exploration of a substantial patch of seafloor like this, and then possibly some physical samples. And you know, John's been talking about box cores and other forms of physical sampling of the seafloor. You get all those sources of ground truthing.
Yep.
Pair that
Go
with
for that,
the
please.
acoustic record, and you can start making some inferences about what you're seeing at a local or regional scale. But for example, you know, if we were mapping this with the vessel right now, each pixel on your screen that I'm looking at in HiPAC right now is probably
Go for zoom.
on the order of 100 meters square. So 5 times the amount of area
That's great, thank you.
that's being illuminated by Little Herc right now. That's 1 pixel on your screen. So the ability to say, you know, you're certainly not seeing individual nodules at that range. We're just looking at broader trends in echo intensity.
Cool, thank you.
And if you use an AUV that's real close to the seabed, you can
Yep.
get a lot more detail,
Yep,
right?
exactly. So that's just you know, you're always fighting your triangles and the laws of physics. So if you bring your sonar closer to the target, closer to the seafloor, the better resolution you get. And in our case, and tantalizingly close to 5,200 meters of depth, we're quite far away. So our ability to discriminate detail on the seafloor at that range is pretty limited. So we're looking for broad trends and then getting down closer with ROVs, AUVs, other technologies when we really want that additional level of detail.
Yeah, so you can map in a qualitative way, but to quantify
Exactly.
the actual abundance, I don't think has been demonstrated convincingly in terms of acoustics, things like backscatter.
Cool, thank you guys.
Yes, the purists will tell you that, you know, a multi-beam like the one on the Nautilus and most multi-beams, they are not calibrated instruments. They can be if you want to, say, stick Nautilus in a giant tank and swing calibration spheres through each beam of the sonar, but that would, it's not an entirely practical exercise. So we sort of live with the fact that it's qualitative. There are some tricks you can play by pairing that with a calibrated sonar, like fisheries echo sounder or a split beam echo sounder,
on that.
like the EK series.
It's an interesting worm. Is it a worm? Or is that
could
just a
be.
cast?
It could be a cast too, but what do you think? Looks like a cast is cracked there. Okay, I think we're good, thank you. Let's have a look at that. No, don't worry about it,
Yep.
I think.
Okay.
Well, hang on. You want to zoom in?
Oh.
Okay.
On the right.
Are these ones?
Oh, there. Yeah. Let's get out of my dust cloud a little bit here. Okay, let's just zoom there.
Let's go for the top of the stem. Interesting, huh?
Thanks, Tim.
from our last watch, we had someone who shared a riddle, and I don't know if we said it on SPL or not, but I wonder if any of you all can get it. I think I know it, but I'm not sure. And the riddle goes, I am on you, but I am not touching you. What am I?
A thought.
There's cameras. They're on you and not touching you.
That's true. I was thinking light. Light's on you, but not touching you. Jaina, do you have any hints? Any suggestions?
Camera's kind of smart.
Yeah, cameras seems like it might make sense. Lindsay or Sandy, you want to weigh in on the riddle mystery?
I feel like Jaina's answer is making me think there's like, I don't know, there's that saying that's like, oh, all eyes on you, you know?
Can we zoom on that?
Yeah,
Sure.
eyes could be on you.
Can you zoom, please?
Thanks, Tim.
Thank you.
Thanks, Jaina.
Someone in the chat guessed hair or like something that's connected, but I guess hair is touching you. So it's, I'm on you, but I'm not watching you. What am I? Or sorry, I'm on you, but I'm not touching you. What am I? Yeah, I'm gonna guess light. Cameras or eyes are all really great lights, or great guesses, I think.
There's a more cynical one. Dead.
I
Dead.
was going to say burden, yeah.
Oh, that's funny.
You want to zoom on that one? Are you good?
The general existential
I
crisis.
think she's good. Let's keep going.
Okay.
Oh, I just got some intel from downstairs as well. Sounds like we have a snack identified.
Do you know what it is?
Yeah, do you guys want to know?
Well,
Or do you not want to know?
or should we guess?
There's a lot of guessing today.
We've been getting a lot of cookies recently.
It's not a cookie.
Oh, it's a change.
I heard perhaps we might be out of chocolate, which is sad.
That
It's
is
really
a little
good.
sad.
Rye just brought me some. Thank you, Rye.
Oh, Lindsay, the answer was eyes.
Oh, it's there.
Ooh, okay. Well,
Yeah.
Jaina definitely
I think cameras work
put me
too.
on that. Yeah.
Light, I don't know if light works because I guess technically does it touch you?
Yeah, the photons do.
Photons do.
Try and get all the way there.
Okay, the next riddle, the riddle of our 3 o'clock snack.
Can we look at that? Well, I've guessed some out.
Is that
Looks
the
like that may be
midwater
a
one?
fish coming up.
Oh,
Very slim.
good eye, Sandy.
Has really low biomass down here, eh?
Yeah. It's interesting, right? Because when I feel like our watch at midnight, we just saw one thing after another right off the bat. Is it a faceless?
No, I think it's got a face, this one.
Is it possible to get a zoom on the head? Oh, never mind.
Camera shy.
Yeah.
No, it's out of the light.
Oh, that's another question I
You want to go for the holothurian? Or
Yeah,
the-
let's.
Yep.
Sandy, when
Go-
you
go for zoom.
see multiple things near each other, do you make an observation for each of them?
Yeah, I usually do.
Okay.
Sponge.
Yes, can we get the stalks at the bottom? I think it's worth trying to get a photo, or you stable enough?
Tilt down a little. There we go.
It's like it's on little stilts, isn't it?
Tilting
That's
up.
wonderful. Thank you for that.
Okay.
Yeah, it does. It looks like a house in like the Outer Banks.
Yeah, I feel like on my previous cruise in the Marshall Islands, like most of the sponges we saw had kind of a thicker base, but here they're kind of like you were saying with this one, they're really thin. I wonder if it's...
Get a zoom on the way past. I don't know if we need to stop.
Okay. Go for zoom.
Yep, thank you. I think they've got that one. Where are we? Oh, let's head over to port, I guess, eh? Is that what you want to do?
The fish earlier reminded me. So I've seen a couple of the faceless Cusk eels that you guys have seen. How do those eat? Oh.
Yeah, how do they eat? I have no idea. I bet Sandy knows.
Probably with their mouths.
Oh, so they do have a mouth. Okay, couldn't see that.
We have observed the Cusk eels on a baited lander. So we had some bait in a laundry bag. You know the laundry bags with all the holes in it that you can throw straight in the washing machine. And we left there's a multicorer that we left on the seabed for about 5 or 6 hours, and then all these animals turned up. And the behaviour was fairly consistent. There were rat-tailed fish that would attack the bait, and they imagine that's a corpse. They'd be trying to tear it open, and the Cusk eels wouldn't engage. They would sit back. And so I guess the idea is that the rat-tailed fish open up the animal, and then the Cusk eels can get into some of the softer bits in due course.
A lot of scavenger feeders have the mouths on the bottom of their head, and so they kind of suck it up once they go along the floor.
So what they both would do is, while they're sort of hanging around, there were lots of amphipods going in and out of the bag, and especially the Cusk eels were quite good at sensing when they came very close, and they would suck them in like that. Yeah. So it wasn't like either fish type would go short of a snack while they're waiting for the main dish to be prepared, so to speak. Can we actually go that way? Is it too late? Yeah, we did it at 2 sites and we got identical behaviour at both.
Do you think there's like a... I guess we haven't really been
Holothurian
observing
there, or
them
the white
together.
thing in the front?
Yeah, you want to look at that? Let's do that.
Let's do the white thing in the front.
Yeah.
Tip the back one we probably don't need to zoom in on.
Okay.
It's another one of those wormy things, is it? Okay, thank you very much. And we'll just do a flyover of the Holothurian.
Okay.
We've got the footage of the baited lander on board if anyone wants to watch it.
It's actually very cool. Saw it when we did that presentation.
Another Holothurian.
It seems like all the creatures down here are pretty solitary, but I almost wonder if like those two different types of fish could have like a symbiotic relationship or something. They tend to be near each other.
Yeah, you could see there are also decapods, prawns there as well, and they all seem to be able to coexist. Although the decapods, there's a star, a brittle star, the decapods will be much more prone to being cautious. So I suspect the rat-tailed fish would snack on them if they got half a chance.
I heard the other day on deck some of the ROV engineers like talking about the placement of the different lights. And I was wondering how you guys decide that. Do you like do the first dive and then make adjustments, or yeah, how do you figure out what it's gonna look like?
Yeah, we do best guess on the first dive and then see how they are and evaluate it, and then we play rock, paper, scissors to decide who gets to adjust it next time. Yeah, no, we all kind of look at the video and try and decide to move this one up a little or move that one out a little, but it would definitely be easier if we had small pan and tilt units on the ROV, but being a smaller ROV, we don't have space for pan and tilts on the lights.
Let's head towards that one, please.
Okay. But yeah, rock, paper, scissors is generally the answer.
Nice.
But we invented a new version of that at the beginning of this cruise. What was it? Sponge. What was it? Rock, sponge, megafauna?
Maybe nodule sponge fish or something, I don't know.
Yeah, what was it? Sandy, do you remember?
It was
It's
rock sponge fauna.
Once I get the
Rock
latest.
This looks like a
fauna.
starfish
Zoom in
up ahead. I would love to get a zoom on that.
Yeah.
It's a big one, that one.
Yeah, different from what we've seen
It's
before.
20 centimeters across.
Zoom and I'll tilt up.
That'll be about that big. As big as a dinner plate almost.
I think another watch saw something similar. I saw it in the video or in the photo captures.
Can we get a zoom in maybe on the end of it here so we can really get a profile of those polyps or whatever they are. Yeah.
Tilt up a little.
Oh, that'll do. That's fine. Go for double on that one.
That is double.
That is double. Okay. So we're fully zoomed in?
Yes.
Okay, thank you. We're good. We can move on.
So Sandy, when you're logging, there's some different buttons for like making a biological observation versus like a different one for taking a still cap. How do you a still photograph capture? How do you decide when to do that versus when like the observation photo is enough?
Whenever it looks prettier. I love it when we get to do the doubler. I think it's so beautiful. The camera's incredible. The capacity we have to like really look and see. So if there's like an interesting like detail on the organism, I would love to take like a still capture. But so I never think one picture's enough, you know?
You want to zoom in on that stalk? Let's go with it.
Is it this stalker? Yeah. Okay.
Actually, sorry.
Zoom in.
Yeah, zoom in, but then I'll get you to come back and zoom on something as well. A little holothurian. It's looking perched on top of a nodule. Yeah, it's a parasol one, eh?
Carnivorous sponge.
Yeah, thank you. If you zoom back out again. Oh, let's try a look. Go down, just directly down here.
Might be a little too silty there.
Yeah, that thing there.
I just
So there it goes. Doesn't look like it much. Thought it might have been an animal. I think it's a nodule with a bit of mud on it. Thank you.
It
Lindsay
looked
and
like there's a cast down there or something too. Is that what you guys have been calling sort of the long poop strings?
There's different holothurians leave different kinds of tracks. So I think that circular one, or that might have been the one from the what were those worms called? The
nemertea worms, the nematode worms, or the acorn worms.
acorn worms. I have a feeling that was from the acorn worm, whereas the bundle with a bit of a string is from the holothurians that leave the tracks behind generally. We're not experts, but I know some people seem to be able to match cast with species better.
It's like those like scats and tracks books. Do you guys have those for identifying animals?
That's us.
Totally.
Scats and tracks of the deep.
Did you get an ID on that puffy slime star thing that we just saw? No. Someone was asking in the chat if we knew what it was.
Let's head towards that, please.
Yeah, I'm calling behind Atalanta here. I might have to skip over that one. And,
okay.
And
we'll pick another one up somewhere.
Do you guys do anything for like your eye health while you're sitting here? Do you like try to look away every time?
Hope for the best.
Yeah.
Maybe blue light glasses would be useful.
Yeah.
You know?
I wear blue light sometimes. But yeah, that's a great question. There's a lot of artificial light in here and screen light, huh?
Yeah, I know there's that thing where it's like, oh, every 20 minutes you're supposed to stare into the distance for 20 feet away for 20 seconds or something like that. I haven't been doing that, but my eyes are getting tired, so next time I would.
Yeah, 20 feet away, like, I don't even know. You can't even look anywhere 20 feet without a screen in here.
Okay, all right, it's coming to the end of the hour, so I think I'm going to head back down and see what our mystery dessert is.
Lindsay.
It was wonderful having you.
Yeah, thank you guys for having me and answering my questions and letting me shadow you.
Okay, so I do know now what the snacks are.
Yeah.
Goodbye.
It was a trick. Who's been reading the group chat?
No, I'm good.
I just ate one. They're really good.
Are they good?
It was really, really yummy.
Oh my gosh. Better than the breakfast cake?
I don't know if I've had the breakfast cake.
I've had it every day.
I don't wake up for breakfast.
Oh, that's right. Bye, Tim. See ya. Do you think there'll be some left over after our watch, Jaina?
I don't know. Rye brought me a piece.
Oh, nice. That was good of Rye. Thanks, Rye. What about us?
That's my roommate.
Yeah, I guess you guys are roommates. You have to take care of one another. Bye, Lindsay. Enjoy the snack. Yeah, someone's asking, what are the dots in the middle of the screen? And those are our lasers. So they're centered 10 centimeters apart so that we can basically size different things on the seafloor, because it's really hard to get a gauge of depth or scale from a camera about 5,000 meters away from the ship. So those fluctuate based on how close or how far away we are from our particular object.
Can we do a zoom on that one if we've got a second, Josh?
Yep. Zoom in, please.
Thanks, Josh.
Okay, come wide.
What depth are we at right now?
5193,
Yeah,
still not quite, huh?
Now we're going to climb, I think, from here. If you look at the contours,
Yeah,
we'll go all the way up to 51, maybe 5160, 5170.
okay.
And then we'll drop off the other side.
So we're ascending in elevation, and then are we going to drop back down into another nodule field? Yeah, or?
Well, there'll be nodules everywhere, but as we drop off, I'm hoping, based on the backscatter response, there'll be a lot less nodules.
Okay.
Yeah. This first part was always going to have quite a few, but I'm not quite sure where the transition is based on the bathymetry.
Okay. That makes sense.
Sure. Okay. Okay.
Go look at this little thing.
Okay.
We'll try and get a zoom on this, please.
Okay. You'll zoom in, please.
That's great, thank you.
Okay, come Mike.
You want to go for one of the white flecks? Should we try this one? Let's go for that little white fleck if you've got a chance, otherwise we'll go for another one.
That's all right. We're going to get there.
And on the way we'll look at that as well, that brown thing.
Okay, we'll zoom in.
Oh yeah, little gastropod. Yep, sweet. And then
A little.
yeah,
Oops, come a little wider. Oops.
that's fine.
Sorry, we're getting try again. Sorry.
No, it's good. We'll keep going. And we'll head over these on the way. We don't need to zoom, just nice and close. Yep. You good with those? Yep. Matt, quick question.
Go ahead.
How are we going with regards to schedule?
Right on schedule.
Great.
Yeah, we've maintained a consistent 0.2, so should put us at waypoint 10,
Yep.
right on time.
What time's that going to be?
Should be about just before 6 this morning.
Cool.
Or tomorrow morning.
Just pass over it. Unless Sandy wants to go for a zoom.
Sandy, we have a bio question, and someone wants to know, why don't
That's
we
good, thank you.
see sharks or whales or octopus down here?
Well, not 100% certain why, but a lot of times like mammals, so like whales and such, can't really dive this deep because they will run out of oxygen, or, you know, so sperm whales and minke whales can dive pretty deep,
We'll
about
zoom on
like
that one.
3,000 meters to 2,000 meters, but 5,000 meters is a little bit out of the reach for most creatures that need to go back to the surface. You know, you never know. There could be some down there that dive that deep, but I've never seen any. And as far as, you know, maybe bigger fish such as shark, there's shark-like fish down here, but they're just not very common to this area.
Zoom in, please.
They do better in shallow areas. There's more fish for them to eat. There's more nutrients up there. They can grow larger. So yeah,
That's a
just
big
different
xeno, I
ranges
guess.
of creatures that are better adapted
Amazing.
to
Okay, come on.
Thank you.
Yep.
these different zones in the ocean.
Okay, and a follow-up question. What is the most startling thing you've seen or observed on a dive?
Grab that, hey, Sandy.
Yeah, I got that. Thanks, John. That's a good question.
Would it be on one of these dives?
Probably the little monopsid isopod that we saw the other day. That was really, really cool. That startled me. It was very beautiful.
It was beautiful.
Okay. No, I missed it, I think. You want to go for a zoom for this?
Sure.
Mikhail, do you have
Okay, we'll zoom
something
in, please.
that startled you while you were observing any of these dives, or a different dive?
Sorry, your mom.
Not startled, but
There
there
we
was
go.
like that freaky
What on earth is that, do you
lizard
think?
fish that was kind of on the bottom, just laying
Yeah, I think it's a bit of bone. Maybe a bit of vertebrae. Hasn't dissolved away at all. There's a little critter living inside. You see it? Yeah, yeah. You want to grab that one, eh? Thanks.
Okay.
Right. Oh,
Sorry.
Oh.
Mikhail.
Uh-oh.
Sorry,
Stand
Mikhail.
by.
there. Yes?
I clicked extender and it went black, and now I'm lost control.
Uh-oh.
Okay, I've lost control of camera.
Let me see.
Okay. Power cycle. Hey, camera's off. Power's on the camera.
Hey, there's the seafloor.
we got a authentic view of the abyss there for a minute.
Yeah,
Yeah.
no kidding.
Completely dark.
Yeah, so that's what it looks like normally, huh?
Yeah.
Nice hustle, team. Good job.
So I guess you're right. If it's something that's on you, it needs light to work, doesn't it? If it doesn't want to touch you.
Yeah, there you go, yeah.
Can you zoom on that one, please?
We'll zoom in, please.
Yeah, it's looks like a couple of old stalks or something that's just been colonized. Thank you.
Okay, wide.
I think we just do a pass over that and get a little zoom on the way through.
Zoom in, please.
That's great, thank you.
Okay, we'll come wide.
And let's head that way. Oh, I think it's a crinoid. That's fine, thank you. Yeah, I'll go through there. You want to zoom in either of those, Sandy, or you good? We just fly over them, thank you.
Okay.
We've seen those two guys many times before.
Yeah.
They're very cute. We love them.
Yeah, that's the whole thing. It's good that we get our eye in, right? We do the zooms, we learn what we're looking for, and we get more observations this way. Let's go for this one here, please. Oh, it's a crinoid on that one. I think the other one is as well.
Let's just fly a bit closer to be sure.
Yeah.
Thanks, Josh.
Interesting how those crinoids seem to have a very consistent orientation.
I guess they must turn, do they, into the current
Yeah.
with the tide?
Are you, yeah, can you feel the current pushing in that direction?
I don't feel too much current on this particular dive. I felt it more on the other abyssal plain area that we were in. Here it seems pretty gentle.
I might go for a zoom on that one, eh?
On which one?
That one there. Thank you.
Oh, there, yeah.
Yep.
Oh, zoom in, please.
That's a new one, isn't it? Looks like another
It definitely
sea
looks
urchin.
messier than the ones
Yeah.
we've seen before.
And what's this on the side? See this?
Yeah.
Is it as zoomed as we possibly can?
Yeah.
Awesome. That is a different creature there.
Yeah. Just when you think you've
Sort of
seen it
similar,
all.
yeah, similar branching to some of the bryozoans we've seen, but is that a little baby fish in the foreground?
I wonder. Yes, that
Oh,
thing.
they look like two baby fish. Do you see both of those there? I've seen this in like shallow ecosystems where baby fish will hang out in different structures such as that as little nurseries almost.
Wow.
So that's interesting.
I wonder if it was a bryozoan taken over. Yeah. And
That's
then,
what I thought too. Maybe like a dead bryozoan. Not sure.
The other thing is that at these depths, of course, even silica doesn't last forever. So
Right. Everything turns to dust.
it can. Yeah. Again, the CO2 can dissolve and reprecipitate silica-based life. That's why this place is littered with massive piles of dead sponges. And it actually can be incorporated into the nodules and crusts as opal. So kind of opal cristabolite, opal CT. And yeah, not the beautiful opal that you get in Australia. But it's a much slower and less consistent issue than with a calcite.
Jane, it sounds like we have some baseball fans in the chat.
Yeah, we'll go for a zoom on that, please.
I haven't looked at the
And
score
we'll try and
yet.
get that little critter in the foreground as well, if you don't mind.
Zoom in, please.
Do that, and we'll do the big guy. Extender work that time. All right, let's go up here. Look at that if we can. Again, I've got no idea what that is. Wonder if it's a piece of pumice, actually. Yeah, I think it's a. Let's log that as a piece of pumice, and it's just been covered in that stuff.
Okay, okay, my please.
Because it could have been a single piece that's broken into three. That can be quite delicate. Yeah. Where are we? Over here, so we can go that away. Look at that bryozoan on the way. And then there's a holothurian over there, but we'll just keep going on this heading. Looks like a sponge over there. Yeah, let's do the fly past just to try and get the form. Maybe a bit of a zoom.
We'll zoom in, please. It's good there.
Yep, no, we got it, thank you.
Okay, okay, Mike.
Let's take a look at that one, please. Would like a nice good zoom of the top.
Okay. We'll zoom in, please.
That's a cool photo.
Yeah, that is. What are we looking at, Sandy?
Well, okay.
Thank you.
Looks to be like some sort of creature perched on top of a sponge stalk with two little isopods below it.
Okay, that was a sponge then.
Yep.
And we'll grab that as a zoom on the way past. In fact, looks a bit different. That's is it a
Zoom in, please.
yes, please. Let's get that one. We might have to stop. Oh no, it's a shrimp.
Shrimp.
Very
Yeah,
pale one, isn't it? But
come wide.
we've seen that style before with a very long. There he goes backwards. Let him go. Can we go over those mounds? Is it too far out of the way?
No.
Get a zoom on that one, please.
You'll zoom in, please.
It's a different form, isn't it?
Yeah.
Let's try and get a good photo of that one, please.
Okay.
We might
Okay.
need that
So
for the
get settled
catalogue.
down here.
Holothurian?
Yeah, that's a different one again.
Yeah, it's interesting how it looks kind of segmented and it's
Could we
not
get a zoom in on
opaque.
that left end, please?
On here.
Yeah.
That
if you
left,
could,
okay.
if that's possible.
I'll try and slide over a little bit.
We might be able to blow it over, let's see.
Oh, yeah, we're going to get blown over now. Yep.
It's all right.
Okay,
Thank you.
I'm going to
Thank you.
keep moving on here.
Appreciate it. You know how wombats will take dust baths, Sean? Wombats will take like dust baths.
Looks like we have something off in the distance there.
Sorry. Yeah, let's go look at that. Looks pretty interesting.
Have we identified, somebody wants to know the species of shrimp that we've been seeing on these abyssal plains?
No, it would be hard to identify unless you really get into the detail of the different legs and segments and colors, and I have not sat down and done that yet. But they are arthropods, crustaceans.
We'll zoom in, please.
Oh, one of those snails, huh?
Yep, it's quite a large one, isn't it? Because
Yeah.
we'll just double check the nodule size on the way out, but it's about 3 or 4 of the small ones. So if we could zoom out when we're ready.
Okay.
Yep, let's just stop when we see the lasers, stop there.
Oh, wow.
Oh, it's almost 10 centimeters.
Yeah.
Eh? Yep.
So
I can try and...
Nice.
There you go. That'll give you.
Thanks.
So it's more like 5 or 6 centimeters.
Centimeters, yeah.
7 long ways.
So are their
Oh,
shells
come wide, please.
calcium carbonate? Or do they make shells from something else?
Oh, there is some calcium carbonate around. It's in the water, for example. Yeah, because it dissolves. It's and there must still be a little bit, a few percent in the sediment. So they will find some calcium carbonate here and there, especially as they're eating the snow, which may or may not have completely dissolved. But
Sure.
they have a hard time of it, yeah, generally.
Yeah.
They, there's been studies done on the effect of ocean acidification with shell thickness and things like that, and so animals develop adaptive strategies to compensate. Go for a zoom on the right-hand side.
We'll zoom in, please.
Holothurian. That Star Wars Holothurian.
That's right, the Imperial Walker one.
Yeah.
With the petropod shell behind it. It's great. And a sponge at the back there, is it? We'll just do a flyover and a mini zoom.
Okay. Yeah, we can do a partial zoom. Fly over. That's good.
Yeah, thank you.
That's great, thank you.
Appreciate
Get
Okay,
going.
come on.
And we'll go over that one again, I think. Actually, we'll do a zoom on that one, please.
Okay. Whoops, we'll zoom in.
Probably an anemone.
Yeah.
Very cute.
Very
Thank
good.
you.
Yeah, we'll come wide.
You good with that one, hey?
Yeah.
Cool. Oh, I missed something there.
So I see we're just sneaking up on waypoint 5. Did we have 10 waypoints altogether? Is that what I saw?
Let's do a little zoom on those.
That's correct.
All right, so we're just about halfway through.
Hey, we'll zoom in, please.
Oh, yeah. Anemone. What's behind it? Actually, another gastropod, I think, eh? Yeah, I think we've got enough for an ID, thank you.
Okay, come on, please.
Oh gosh, the patterns of the seafloor are starting to play tricks on me.
Can we just have a quick look at that on the way past? Don't know what that was. Maybe we should have gone for a zoom. But oh, it's one of those bits that are stuck in again. A bit of crust that's vertically embedded in the mud. Oh, we should have zoomed on that. Is it too late to go back?
No.
I think it's... wasn't that?
Yeah, I think so.
Zoom in, please.
Yes. Either it's a cast or another one of those strange worms, but I think it's a cast, eh? Yeah, it's not, that thing hasn't moved, it's covered in stuff. It's definitely a cast. Thank you, we'll keep going.
Don't mind.
It's a worm-like cast.
Not a holothurian though, one.
No.
Different source.
Yeah, let's head towards that.
Looks like a fish.
Mm-hmm.
One of those Cusk eels.
Turned and wondering what we are.
What do you wager, eyes or no eyes?
It's looking at us.
Oh.
Oh, that's another barnacle, I think, down below.
Oh, yeah.
Oh, that floater is.
Could we get
Zoom in,
another
please.
zoom on that Triflonus nassus?
Yeah, look at those little eyes.
Faceless
Yeah,
Cusk eel.
and you
And
got
a jelly,
next
yeah.
Yeah.
Beautiful.
Oh, look at that.
Wow,
That's
you can see its
incredible.
mouth.
Yeah. Underneath.
Yeah.
That's the best angle we've seen of one of those.
Yeah.
That might even make a highlight, eh?
It might. It sure might.
That's all the tilt up I have.
Thank you so much, Josh.
Okay.
It really does look like a tadpole from that view, doesn't it?
Okay, we'll come wide,
Awesome.
please. Or I guess we are wide. Okay.
Great. Yeah, let's go that way. you want to choose one, Sandy? Thinking they're
Um,
both holothurians, I think, aren't they?
yeah, let's go up to that thing in the center far.
This one?
Nope,
Oh.
center far.
This one?
Nope,
Or up the back
left.
there?
Yep.
Yeah, okay. Sorry, this one
That's
here.
okay.
That's all right.
We were
Yeah,
just
I'll do a
working
little,
out where we're
do a little
going.
flyby about all these.
Well, I think we've got the two holothurians at the
Okay.
in the bottom field of view.
And I'll hit that one little blob, and then I'll cruise over
Yeah.
to the left.
Yep, that one there. I think that's a
That's an urchin.
yeah, and there's another urchin. And we'll oh, that's interesting colour.
Yeah, it is,
Go
isn't
for
it?
that.
Kind of green.
Right.
See if we can get a good photo of that one, please.
Looks like one of those, looks like an enemy or hydroid colonizing a sponge stalk.
Yeah, if we could swing to the left and try and get a view from
There's a bryozoan on the
sort of like a
bottom
view
there.
like that, that'd be bonus points, but don't worry too much. Great, that's a good angle.
Kicked up a little dust there. Okay, we'll zoom in, please.
An anemone?
Or colomorph, because of the mouth in the center. It only has 2 rows of tentacles around, and it sort of protrudes up.
That brightly coloured thing's its foot or something, isn't it?
Okay,
Okay,
come
thank
wide.
you.
Yeah, it did look like it had a unique color from afar.
It's underneath it, yeah.
We are at a depth of 5,191 meters, for those of you asking. Just shy of what we thought was going to be our max depth with Little Hercules ever, which would have been 5,200.
It's a bryozoan, I think, there. And a holothurian there. And a couple of teeth there.
Couple nodules I see.
Yeah, 1 or 2 trillion. There's lots of teeth, eh?
Oh yeah, like that down at the foreground.
Yeah, we'll head over that guy. I think we'll just get a zoom on that quickly as we go over.
Okay.
Actually, stop and let's do a proper zoom.
Zoom in, please.
Huh.
Sponge.
Interesting, isn't it? Thank you.
Okay, we'll come wide.
Should we look at a small flick? We'll go for that one, please.
It'll zoom in, please.
Yeah, tetrapod shell.
Yeah, possibly one of those.
Got that one straight away. If you zoom back out, there's a couple
Okay.
to the right. I don't know if we've got a chance of looking at them while we're here. There's one there, and there's one just below, I think. There. Whichever you're comfortable with.
Okay,
The one on the right actually looks a bit more interesting.
turn a little bit. Sometimes when I turn, I'm going to kick up some
Dust, yeah.
That's okay.
That's all right.
dust there.
What we can do... Yeah, let's go for that one. What do you reckon that is?
Sort of has the translucent interior. Very nice. Thank you,
Okay,
Josh.
we've got it.
Okay.
Can we get that one while we're here?
We can try it, yeah. Go ahead and zoom.
Yeah, and another
Yeah, no,
tetrapod shell. We're good. We can go now.
Okay, come on, please.
Pteropod, sorry.
Oh, no, you're good.
Yeah, I know.
Possibly. There's also different pteropods.
Yeah, she put a question
as
mark,
they
didn't she?
die, yeah. Their shells could maybe fall to the ground.
Bryozoan.
Yep.
Someone is asking in the chat if we can take samples. Not on this cruise, or on this dive rather, because we're using Little Hercules, and Little Hercules does not have any sampling tools. We actually just have one big MXD camera, which is the Monterey Bay Aquarium Research Institution and Deep Sea Power and Light super duper high def camera. So we're doing visual surveys of the seafloor. But when we're diving with ROV Hercules, we can take samples. So we have lots of different sampling technologies. On ROV Herc, we have 2 manipulator arms, Niskin bottles, push core samplers, all kinds of things.
No, just keep it going. So it's a bit like the answer to your riddle, huh?
Yeah.
Yeah,
Exactly.
what's on it but can't be touched? Seabed fauna during a Little Herc dive.
Totally. And yeah, the reason that we're using Little Herc is because we are diving at such extreme depths. So right now we are at 5,191 meters. Little Hercules has a max depth capacity of 6,000 meters. Hercules I say just 4,000 meters, but it's still quite a bit, 4,000 meters. So that using Little Hercules, we can get down to these really, really deep abyssal plains out here in the Cook Islands and see what's going on. Have you been surprised by this dive at all, John, or are you seeing what you kind of anticipated?
Well, we're hoping to find an area with less nodules in it, and waypoint 6 was kind of the place where we most expected to find it. But the data we're using is a little bit imprecise. There's a good holothurian, yeah. Look at that track, that sediment track. I wonder what did that. It's almost like something got dragged through. So yeah, I was hoping we'd get to that point kind of on our watch, but we're on schedule. So I guess the next watch will probably get to waypoint 6, and as we cross that little ridge, hopefully there'll be an area with a lot less nodules. And we want an area with less nodules because we want to compare the megafauna on a place with less nodules with an area with lots of nodules. And funnily enough, this is a hard place to find an area with not too many nodules. But we were also constrained a little bit on where we could place the dive. So at least we're getting some good data now.
Yeah.
On what's found in this particular part of the nodule field. Yeah, that's...
Let's go up to that. Oh, never mind.
We haven't seen a track like that before.
Yeah, it's interesting because it, like you said, it almost looks like the sediment's on top of the nodules rather than just kind of that dusting.
It can't be a drag device because it's got those loops. There's definitely an animal that's gone around in circles. And normally the holothurians do the opposite, don't they? They take the sediment off the nodules.
Yeah.
So.
Be about 3 hours for the vehicles to get to waypoint 6, John. So
yeah.
Right after dinner, you can tune back in and see what's up.
So is that about 3 hours in between waypoints is kind of what we're
Yep.
pushing for?
Thereabouts. The ship is passing the waypoint now, and the vehicles are about 130 meters behind us at the moment.
Oh.
And that's about 20 minutes at our current speed.
Cool.
Can we grab that? Sorry, the last second. It's down there.
Zoom in, please.
Okay, thank you very much. When we come out, I'd like to look at that side. There's something wandering the seafloor. There it is, fella.
That same little arthropod.
Let's see if we can get an image of it.
Just scooted into the frame as we were settling.
Very good, thank you.
Okay, come wide.
Chat would like to know, has anyone ever found a paleo nodule field?
Yes, in the Pyrenees, which is a mountain range between Spain and France. There are some of the rocks there are what Jurassic age, and people have found nodules in some of the sediment beds there. So that used to be part of an ocean floor, but when plate tectonics meant that Africa crashed into Europe, the Pyrenees were formed and a bit of the ocean floor was pushed up into a mountain range. And someone found some nodules in that once. You might zoom on that one, please. Oh, too late, don't worry, it's okay. So the formation of polymetallic nodules requires oxidation of manganese and iron that's in the seawater, so it comes out. So you need an oxygenated ocean, and the Earth's about 4 billion years old or so, and if you can zoom in on that one. The oceans were oxygenated, we believe, from other rocks, not from nodules, about 500 million years ago. So it's theoretically possible that you could find older nodule fields than the one in the Pyrenees.
Okay.
Which do you want to zoom?
It's down below us now. I think it might have been a bit late, or I think it was those. It's pretty hard to see.
Zoom in, please.
Yep, okay, so that's one of those ones, yeah, but getting overgrown. We've seen a few others with more overgrowth on them.
Okay.
I suppose the danger of working for a carnivorous sponge as a cleaner.
I guess so, right?
You get eaten.
Yeah, it's a little risky. Toxic work environment.
And then no one wants to work for you anymore and you get covered in that stuff.
Well, we're just getting ready for our watch change, so I can hear the 4 to 8 folks coming on in. I have one more question for you, John, and then maybe we'll kind of shuffle off and let the new team come in. But is there a way to determine how old those tracks were, those sediment tracks that we were seeing just a moment ago?
Not really that I know of. When the track is fresh, it's got crumbs and lumps next to it that would tell you it happened 5 minutes ago, something like that.
Guess what?
And then eventually those will break down, maybe through currents or from other animals having, filtering through them. So they look pretty old, but that's a very subjective estimate.
Sure.
Yeah, and I wouldn't be able to say it was, 23 years and 6 months ago, or if it was Wednesday last week. But yeah. And I guess if you look at enough tracks, you kind of get a feel for
Sure.
that. That was the first time we ever saw that kind of track. All right, I'm signing off, guys.
All right, enjoy
Thanks,
the shortbread snack.
Yeah.
And thanks, everyone. I'm going to pass it on over here to our 4 to 8. But enjoy. The 4 to 8 is the octo watch, so I'm sure we'll find an octopus at some point. Yeah, the octo watch, yeah. All right.
Dave's in video now.
Howdy, Dave.
Got a couple of like the brief thruster pickups for like just for a couple seconds and then they come back, which I think is like a comms issue. But we had one where Tim lost the quarter for like 2-3 minutes, and then it came back. So we had that issue too. When that happened. Whoa!
There it is, Octowatch.
No kidding. Look at that.
All right, we're gonna get some highlight reel stuff here.
I am so excited. Oh my gosh. This is incredible. Wow, wow, wow. It's an octopus. This is so, oh my gosh.
Okay, I'm gonna try and settle down here
Wow.
on this side of the frame, and then just let it cruise on in.
I'm sitting here for this. I'm not leaving this room right now. Wow. Look at it just drift. Oh my goodness. Oh my gosh, is that a Dumbo octopus? Using its little
my goodness.
And I tried to be so gentle.
We haven't seen one of these on the dive so
Wow.
far.
No. Chase it.
Good
We
job.
can only go so far, but
Yeah.
that's incredible. Holy.
Wow. Look how graceful
Yeah.
it swims.
Thank you.
Oh, yeah, thanks for stowing the lasers.
Beautiful.
Oh, I love the black background. That's
Beautiful contrast.
Oh my
The
gosh.
background.
It almost looks like sort of pale pinkish, like a pearl.
The Florida
color.
A-Watch just came on board, and
God.
here
A-Watch
we
is
go.
lucky.
We get to
We
start on a good
always-
foot, yeah.
We always see our...
Or 8
Wow.
feet.
I could watch this for the rest of my life.
Wow.
Wait, so why can't we
We're
just
so
follow
lucky.
him perpetually?
Right, through the whole ocean.
Lead me to your friends.
Oh yeah.
Oh my goodness.
Oh my
Yeah,
goodness.
no, we gotta tell Miguel we need this.
Come on over. We need octopus film reel number 2.
You can, if you want to zoom it all, Dave, go ahead.
I know. A poster.
Won't do too much. I'll
Love
just
it.
fill the frame a little more.
Okay, yeah, I'll make it easier for
Yeah, yeah.
Nice.
Look, you can see
Focus.
it's-
me. Can you,
it's
hey Ryan, can you change your
Open
heading so you can
up.
put little heart?
Beautiful.
Oh my goodness.
Jeez.
Oh my goodness.
See
Wow.
it's gonna
Bye-bye.
go see you later. Wow.
Oh my
What a
goodness.
treat.
Oh,
Yay.
I'm
so
speechless.
epic.
I'm stunned.
I hope
So
you all
put
were
your
watching
auto
that at
heading back
home.
on.
That was
Auto
impressive.
heading on. Auto
Josh,
heading on.
Josh, let it go out of frame.
And now you can turn.
Let it go out of frame.
Wow.
Yeah.
Wow.
That is
That-
the best way to start a watch.
All right, we can go home.
Yeah, I know.
We're
Okay,
good.
I'm done. See you later, guys.
That's the clip right
That was
there.
crazy.
I'll come wide.
Time to go to sleep
Okay.
now.
I like ran. I saw it in the kitchen, and I like ran up here to record.
That was amazing.
That was incredible. Nice job, team. Great imagery.
Yeah, great work.
Whoa, what's happening?
Okay, so now you can go back to
What
what
we
your heading,
were-
I don't know what your heading was on before.
Well, I don't think the rest of
We're on
the
090
ship
or
can beat that.
Yeah. We're not really in competition, but
But technically your watch hasn't started yet.
that's true. But we're here. We're here early. They left.
Yeah, is that where you were on before, Ryan? 090?
I think so. It looks like that's following the ship's track, right?
Yeah, I think so. Okay, cool. That was awesome.
Yeah, nice work.
That was crazy.
Get to have a sequel version of the octopus footage. Part 2.
Yeah, that was probably the best footage we've had yet.
In action, for sure.
Identification.
Awesome.
Gripatoothus.
Just chalk up another Josh Dave highlight reel.
Yep.
There you go.
Yep.
Good
There have
Josh.
been many over the years. Not our first Dumbo octopus.
That is true.
Well, I'm still getting set up back here, but we have the 4 to 8 watch coming on board. I'm Amanda Demopoulos from the U.S. Geological Survey, based in Gainesville, Florida. I'm a benthic ecologist, and I'm keen to get more footage of not only octopuses, but things that live on the seafloor. I have some great company back here. If they're on the comms, you want to introduce themselves?
Absolutely. My name is Michael Parrish. I am a marine scientist with the Ministry of Marine Resources based in the Cook Islands. And I am really excited now that we are starting to watch at the most opportune time ever.
I'm Kahena Wilhite. I'm with the University of Oslo, studying the deep sea and minerals in the Cook Islands. This has been an awesome watch already, but I'll be here answering all your questions. And yeah, my job here on board is mainly meeting with classrooms and talking about what we're doing here on the Nautilus. Can we have
you want me to start in the front?
Sure.
Yeah, so I'm Jacob Stock. I am your navigator, and I work with the University of South Florida as a hydrographer. And super excited. So was that a Dumbo octopus?
It did look like a Dumbo octopus, but I've got to double check.
I mean, we've been joking about it for so long, as if like we'd never find one.
It was.
Yeah, that's crazy. So, yeah,
wow,
great start to a watch.
A big Dumbo.
Yeah, hey everybody, Josh Chernoff. I'm the RV Operations Manager at OET and currently piloting Little Hercules from Vancouver Island, British Columbia. And RV team is doing 12-hour watches, so we get to play games with everybody and find all kinds of cool stuff. So pass it over to Tim.
Tim, you're muted.
I think you're...
Or
no, not muted.
Not an SPL.
Oh, SPL.
The orange button.
There you go. There we go.
Hi, my name is Tim Bowman. I'm taking care of Atalante right now, just keeping sure the height above Little Herc is okay. And so just part of the ROV team doing 12-hour watches with Josh and Ryan's just stepped out for a little break. And yeah, I'm from Vancouver Island as well.
I'm Dave Robertson, Senior Video Engineer for OET. I'm sitting in the video seat. Get to zoom in on fun things like we just saw. I am not from Vancouver Island, but have been there many times. I'm from Anchorage, Alaska, and other points in between. I have a semi-nomadic lifestyle that takes me all over the place. Mikhail, back there?
Yes, I am. I am Mikhail Rosashansky. I am here on the behalf of Deep Sea Power and Light as an electrical design engineer, and just kind of, making sure everything is going good with the MXDC cam. Yeah, just monitoring telemetry, video, and also, clipping any highlights as well on our end.
Well, I was able to rate it a 5 out of 5. Yeah, still not over the Dumbo.
We got a 5.
We got a 5.
And now we'll go zoom in on this creature coming up.
Dumbo octopus very closely followed by what looks to be Typhlonus nasus.
Aha.
It's already a great start to the shift.
This is a type of Cusk eel, Ophidiidae.
Yep.
Is this a faceless eel?
It is the faceless Cusk eel. Faceless Cusk eel.
Essentially blind, They do have eyes that are underneath tissue that's grown over them. They're called vestigial eyes, so they have no function. So they use various different sensory appendages and apparatus to feel around and sense their food and their environment.
Thank you, pilot.
Okay, come on, please.
Yeah. And a shrimp swimming down there.
Looks like it. It's really long antennae. I think we've seen some friends and family of this shrimp before. I believe it's the nematodes.
So we have some excited messages. Oh my God, I leave the room for 1 second and you guys find a ghost Dumbo octopus. That's probably how all the shift people felt right before this.
Oh, Jana already sent me that text.
No.
Vampirotheus? What was it? Immediately got in and got lucky. That was amazing. So glad I got to see that with you. Octowatch. 4 to 8. Forever the Octowatch.
Yep. 3 for 3, man.
Rima Brown said, oh my God, that was definitely the coolest thing we've seen.
Hi, Rima.
Hi, Rima.
Thanks for joining us. We miss you.
Glad we could be your entertainment while you're still at the office, if you haven't already left.
That's really neat how the octopus was using little hairs on its tentacles. I wonder if that was to sense something in the substrate or if it was trying to filter something out of the water. Good observation.
So there are a few different types of octopuses that have been documented in the region outside of the Cook Islands. These include Ceratothus, which has a similar body form, and Grimpatothus. And those are all different than the sort of ghost version. They're more, they have that Dumbo shape to them. And can't wait to hear from the shore side which one they think it is.
We had a question of, do we have any idea as to the length or the size of that octopus? Any guesses?
Bigger than 10 centimeters.
Definitely.
I think it was maybe 20 or so.
Bigger than a bread box.
And then...
Do people actually say that anymore? I feel like that and broken record kind of have fallen out of play.
I hear the broken record one, but not the bread box.
Oh, okay.
I was going to say, broken record is still around, definitely.
Yeah. We have a question. When the ROV followed the octopus that far, does it pull on the tether or do you stop before you get that far? How do you know when to stop or turn?
Yeah, I was fortunate enough that it kind of hung out close enough that I wasn't getting pulled, just barely. And then it kind of held that position. And then when it swam a little further, I just stayed in that one spot and let it drift away. But yeah, have only so much reach that we've got, but fortunately I hung out long enough to get some good video there.
Glamour shots.
Yeah, that was great. We got to be so close.
It was its day to shine.
So we just passed waypoint 5, so we are officially halfway through the dive.
Are we there yet?
We're halfway there.
We are there.
We are halfway there.
5 kilometers to go. and we are on time to finish up in 36 hours.
Well, maybe we need to slow the ship down.
Yeah. No, we got to get more dives in.
Oh, okay.
Still got 2 more dives.
Yeah, we're thinking.
I feel
Looks
like we're seeing similar animals as we did when we left watch.
Maybe a little fish suspended right there.
Yeah, it looks like a fish coming into view. Take a look at that.
Either that or a really big polychaete.
Wow.
It might be.
The seafloor looks really varied in terms of
zoom in, please.
nodule size and very irregular shape.
Oh, it's a
Ooh,
polychaete.
someone called in. Go, Mike. Woo woo.
It's bound to happen eventually, right?
You've got it. You've got the lock.
Oh, cute.
Beautiful polychaete
Okay.
going out of view. This is the Acroceridae polychaete annelid.
Is that a glass sponge down there too?
Yeah.
We have a question of deep down. Is anyone secretly hoping for Cthulhu or some previously thought extinct species?
I mean,
Anyone?
we were talking about Megalodon just, was that yesterday?
That's today.
That was today. Oh, there we go.
I would be keen to see a 6-gill shark.
I would be ecstatic.
I don't know the deepest record for a 6-gill shark, so maybe our shoreside assistants can help dig into that. But I've seen them in the Pacific, in the deep, and relatives of 6-gilled sharks, different species in the Atlantic. Can we look at this? It looks like a black coral pilot.
Max depth for 6-gill shark, approximately 2,500 feet. 760
You don't want
meters.
the people from the shoreside to help us out?
Oh, I definitely want them to help us out, but...
What was the deepest?
760 meters.
Zoom in, please.
Wow, that's a lot shallower than I expected.
That is a lot shallower than I would have anticipated.
Yeah, I don't know about that.
I think we've seen them deeper than
I
that
think you're right,
ourselves,
Josh. Yeah.
Yeah.
And that is where the shoreside scientists definitely come
There
in
we go.
handy.
Yeah. All right, we're zooming in on a black coral, Antipitherian. And we've seen the relatives of this one throughout the dive. So it seems relatively common in terms of the type of corals that we're seeing.
Okay, come on, please.
And I believe the genus name was Abyssopathes. It has sort of a basket shape. Tina Moldoslova and Sako were both really helpful in giving us IDs of the black corals earlier.
For some reason, my science portal is not coming up.
The chat.
Yeah.
Yeah, it's sort of taken a little while.
If we've got a second, what is this?
Oh, yeah.
Other than probably just a rock.
Well, I know that we're looking at pilot.
A blob.
That's
zoom in, please.
Dead sponge.
It looks sponge-like.
Is that a little worm on the side? I don't know.
That would have been a big sponge.
It could be a mound created by something underneath. It's interesting that it looks like it's got some structure to it.
There you go, come on, please.
Looks like we've got a big ridge off to the starboard side of Adelante on the sonar.
Oh, yeah.
So there's a bit of a rise. Well, maybe we'll start to see more varied seafloor coming up.
How far apart are the rings on the sonar?
20 meters on Atalanta.
Okay, so yeah, 35 meters away, 40 meters away. We won't see it.
So Josh, I hear you've encountered the Titanic.
What?
Was that you?
I've encountered Titanic? No,
Oh,
I've never.
no, was it someone
I
else?
have not dove on Titanic before.
That was Todd.
Oh, it was Todd.
That's the Titanic.
Darn, I'll have to ask him about it.
Tim? Yes.
Yep.
Yeah.
Tim
Worked
has
on
too, yeah.
Titanic as well.
Looks like another black coral
Yep.
passing over.
Well, what have you seen that's cool, Josh?
What have I seen that's cool?
Jumbo octopus.
What can
Yeah.
you match to the Titanic?
I don't know.
Percentages,
Not much.
seastar?
I don't know. I've seen some things.
I think the Dumbo octopus was pretty good.
Dumbox was
Yeah.
pretty good. I like, I love all the biology, but I also like working around, you know, volcanoes and hydrothermal vents. They're quite challenging to fly around, definitely in more
not-
expansive vent fields.
Not melt your sensors.
And not melt your lights as well. Sometimes, you know, you have to sacrifice these things, you know, occasionally.
In the name of science.
Yeah, you just get, I don't know, those views never get old. I've done a lot of wrecks as well, which are always interesting and challenging because there's, you know, it can be dangers there that you can get hung up on. So navigating through those are difficult. I've taken small ROV inside a ship before.
Oh, wow.
And was able to find our way into a munitions store and count a bunch of munitions that were in there. So
That's got to be really tricky to not get hung up on things.
That was tough, and we got out, and yeah, it was fine. Yeah, I don't know. Done lots of cool stuff. You know, I've done a lot of cruises on Nautilus over the years, and all of them are super fun. I mean, the coolest part is just the vast majority of our dives, no one's ever seen the seafloor before, and in a lot of cases, well, they've never seen again. So even that alone is pretty cool.
Very true. I feel like there's always new discoveries, and especially on this dive, since it's the deepest that Little Herc has gone.
Yeah, that's pretty cool. Saw a lot of great sharks in the Caribbean area. A lot of hammerheads were really cool.
Out of curiosity, did we make it down to the 5,200 meter mark?
I don't think we quite reached it, as far as I know.
We were 2 meters shy.
2 meters shy, 52?
Yeah.
Okay.
I would love to see a hydrothermal vent. Is that what they're called?
Yeah, they're really incredible.
They're very
look
spectacular.
awesome.
Yeah.
Yeah, and I guess
Should
when
keep
you're...
your ears open for opportunities in the future, Kahina, yeah.
And I guess when you're putting the temperature probes in, you've got to get real close to the hot water. So
You sure do, yeah.
Yeah.
Yeah. Yeah, we put a lot of experiments out onto some of the vents and temperature probes, like right in, you know, 350, 400 degrees seawater. It's pretty wild.
And that one was a carnivorous sponge, the yellow one's correct?
What depths do you find those at?
Yeah, that's correct.
Might be a good question for Amanda. Pretty varied from what I've experienced. You know, they can be 1,000 meters to, you know, 3,000 meters. Just kind of depends. From the ones I've explored anyway.
Yeah, I would say that's a more interesting dive than Titanic. Titanic's interesting, but hydrothermal vents, you can probably see things that nobody's ever seen. And
Chemosynthetic organisms thriving.
Even the ones that we've been back to several times, like the Endeavour field off of Vancouver Island, it changes all the time.
Yeah.
You go back and one whole section that was a giant set of spires or something like that has just collapsed and is gone.
Oh, wow.
And there's a new vent that's opened up over here, and a whole new set of life around it and that kind of stuff. So they're different every single time.
Yeah, I would absolutely love that. Do you guys, when you look for those, do you return to it? Like you'll go one day and then do another dive, or you just pass through? What does that look like?
Kind of depends on the objectives. Endeavour I've been to many times just because it's part of Ocean Networks Canada areas of study and a marine park. And there's a lot of experiments and a lot of science experiments going on.
Yeah, undersea observatory. It's really amazing network of systems and sensors.
So those ones we've returned to several times, and they're well known.
Can we zoom on that, please?
Yeah. Zoom in, please.
A glass sponge?
Yeah, it looks like it.
It'll come like.
Does the color of the sediment look different to you? Like a little darker? More brown than light sand? When we come in, it seems like it's a little
I would say, yeah. The stuff that we saw earlier in the dive was highly reflective.
Yeah. Jacob, are we heading into the trough or out of the trophy area, or?
We are heading out.
Okay, interesting.
Currently. Yeah, we're going up.
Wonder if there's some sort of variation in the sediment characteristics responsible for the color change.
Someone said underwater lakes. Hydrothermal vents fascinate me. I try to watch every dive.
Yeah, have you ever dove on a brine pool?
Yeah.
That would, yeah, that would
the
be
golf.
neat too.
Yeah, super cool.
That's on the list of things I'd like to do, but
we put Herc like right on it.
Yeah.
You could see like the edge of the whole pool. It was, yeah, it was pretty
Created
wild.
wakes.
Yeah.
Yeah. These are basically hypersaline, high salinity lakes underwater.
Oh.
And they occur in the Gulf, and essentially they've done this